last time I basically talked about the lungs and how air, ventilation, and how air is able to get onto the respiratory surface, the alveolar surface, right? Well, today I'm going to begin to talk about what happens next. Once that air is there, we need to have an exchange with the blood, and then we need to transport the blood. Transport the gases, the oxygen in the blood. So how does this work, this exchange? We have deoxygenated blood, venous blood, coming from where? Where is this blood ostensibly just leaving before it's coming here in this picture? This deoxygenated blood is coming from, yeah?
Oh, it's coming from the pulmonary trunk?
Yep, the pulmonary trunk, which is exiting the right ventricle. So in this blood, we have partial pressure of oxygen of about 40 torr, and a partial pressure of carbon dioxide of about 45 torr. Well, this world that we live in, we walk around in and breathe in here at sea level, or roughly so. The partial pressure of oxygen is about 100 torr, or millimeters of mercury, and carbon dioxide is about 40, although it is rising. So because this venous blood has a lower concentration, lower partial pressure of oxygen, there's going to be a net diffusion of oxygen into that blood across the respiratory membrane. And likewise, since the venous carbon dioxide is higher than the atmospheric concentration of carbon dioxide is going to want to diffuse in the opposite direction, all right, across that half a micron membrane. So as a consequence, the arterial blood that is leaving the alveolar surface is going to have, it's going to be in a rough equilibrium with the concentration of gases in the environment, in the air. So its partial pressure of oxygen is now 100 torr and 40 millimeters of mercury of carbon dioxide. All right. So this blood travels back to the left side of the heart, where the left ventricle is going to forcefully pump it out to the rest of the body, to the epithelial cells, the fat cells, the fibrocytes, the muscle cells, all the cells in your body that are metabolically active and need oxygen. So that incoming blood is roughly 100 or a little less millimeters of mercury, and the carbon dioxide level is about 40, still reflecting, this is the arterial blood, reflecting the concentrations in the environment. Well, those gas concentrations here, and this is for a person at rest, mind you. A person at rest, their partial pressure of oxygen is about 40, and carbon dioxide is about 45. So we're going to have the opposite exchange that was being driven by the gas gradient at the lungs. Yes, Brandon?
Where did you say this gas exchange was taking place again?
At the periphery. It could be anywhere. This could be in your muscles. It could be at epithelial surfaces, anywhere that blood is being delivered to tissue. Okay? Anywhere oxygen is being delivered to tissue. And so this exchange happens such that the venous blood, that's part of the venous return here, is going to reflect the partial pressures of gases in situ, right? So we're going to have now oxygen is going to get drawn off the blood, bringing it down to 40, and carbon dioxide is going to be picked up by the blood, bringing it up to about 45 torr. Yeah. during exercise, and I may get to this today. During exercise, this number here could actually go down significantly as oxygen demand increases and more oxygen is taken up. So this can go, if you are in a sprint, for example, you're in anaerobic, you can send that down to maybe 20 millimeters of mercury, and we'll see what impact that has on saturation dynamics of hemoglobin in a bit. All right, so hemoglobin. Hemoglobin is a tetrameric globular protein in the globin superfamily that has incorporated into its superstructure a porphyrin ring, which has an iron coordinated in the +2 oxidation state, coordinated at the center of it in each of the 4 subunits. It is this iron in particular that is going to be binding the diatomic oxygen gas molecule that has been dissolved into the blood. And of course, we know that iron likes to bind to oxygen, as you can see in that cool progression of a sculpture there that rusts with time, although that is a different chemical reaction. Every red blood cell has, the number I have here is 280 million hemoglobin molecules. That's pretty staggering. That's pretty staggering if you think about how many, at any given time, how many red blood cells your body has. That's an enormous number of hemoglobins. Each hemoglobin itself has the ability to bind 4 oxygen. So quite a capacity.
Are they usually saturated with oxygen?
Wait, just you hold on. The story's coming. Yep, we'll see. So what exactly happens when oxygen binds to the heme group? Well, it forms, it binds the iron, it coordinates to the iron, but because of what's called resonance stabilization, the electrons are able to rearrange themselves and form what is called the superoxide ion. I see some smiles back there. Maybe you're remembering organic chemistry from back in the day, or whatever it is you're remembering. Yeah, there it is, resonance stabilization. It is this resonance stabilization which changes the oxidation state of that coordinated iron. And by so doing, it's going to change the electron distribution, the electron cloud around that iron. And when that happens, we have here the unbound iron is slightly out of plane with the porphyrin ring by about half an angstrom. And when it binds to oxygen, diatomic oxygen, and forms the superoxide anion, it changes the architecture about that porphyrin ring. When that happens, that so subtly shifts the architecture of the entire chain of hemoglobin. So I told you there were 4 subunits. There's 2 beta chains and 2 alpha chains. We'll see that more in a moment. It shifts the architecture of that beta chain so subtly by tugging on this histidine, and that histidine is then creating a very subtle conformational shift in the beta chain of the hemoglobin. We're going to see what effect that has in a moment, but it's going to lead to what we're going to call allosteria. All right, this is the molecular underpinning of allosteria. So this conformational shift is not it's not only felt by the subunit, the beta chain, for example, that it's found in, but that conformational shift is propagated and affects the relative conformations of the 3 other subunits in the hemoglobin. Okay, and by so doing, I'll just give you a little bit of the punchline now. By so doing, is going to affect the ability of those other subunits to bind oxygen. All right, so the binding of oxygen in one site is going to affect how readily oxygen is bound in other sites. So we're not going to get just a purely linear relationship here. All right, so allosteria. The definition here, alteration of the activity of a protein through the binding of an effector molecule at a specific site. All right, this can be, this effector can be the binding of oxygen at one of the heme groups, but we're going to see other examples of effectors that affect, that alter the function of hemoglobin in a little bit. So we have deoxyhemoglobin and oxyhemoglobin right here. And so each of these are color-coded chains. This perhaps is beta, alpha, I don't know the convention here. But we see embedded in that, in black, in the space-filling model, we see the porphyrin rings. So if we were to compare the structure, the superstructure, of a fully saturated, or when I say superstructure, I mean quaternary structure. Does everybody understand when I say, does anyone not understand that term? Quaternary structure of the fully saturated oxyhemoglobin. So there are four oxygens bound here and none bound here. The relative orientation of these beta chains with respect to the molecular axis are rotated by 15 degrees going from here to here. So there is a 15-degree conformational shift in going from deoxyhemoglobin to oxyhemoglobin. One of the things you're going to notice, I'm going to draw your attention to the fact that hemoglobin looks like a donut. Right in the middle of the hemoglobin, where these 4 subunits bind, there's a little hole. There's a little hole there. We're going to talk about the donut hole in a couple slides and what happens there. But that's a nice little nook right there that we're going to be able to slip another effector in, 2,3-BPG, which is going to affect the functioning of the hemoglobin. All right. So over here in deoxyhemoglobin, there is this rotational shift which opens up that interior of the hemoglobin slightly, just ever so slightly, compared to the oxyhemoglobin. All right? Yeah.
For the oxyhemoglobin, is that when it's saturated with 4 oxygen, or can that just be 1?
One oxygen is not going to give you the full 15-degree shift, but it is going to shift it a little bit somewhere along that spectrum. Okay. All right. So here are some GIFs that I have that can show you the conformational shift that you see going from oxyhemoglobin to deoxyhemoglobin. So one is along a central axis and the other is on a lateral axis of this protein. And as we shift from deoxy to oxy, we can see that there is indeed a change in this cavity that I was pointing out earlier in the donut hole. We can also see a change in the orientation of that histidine that is associated with the iron, the either +2 or +3 oxidation state iron that is embedded in the porphyrin ring of the heme group. Okay, so it's called heme when the iron is in it. Porphyrin ring is everything but the iron. That's the naming convention if you're getting confused, if it seems like I'm using those words interchangeably. All right, and then here we can see oxygen coming down, and as it comes down, we have, this must be from a molecular dynamics simulation or something, as it comes down, we have a whole host of conformational changes that are going to happen. And they may seem subtle, but they have profound impacts upon the ability of other oxygen groups to bind at other sites in the molecule. Okay. All right. So here is your first introduction to the exciting world of hemoglobin saturation dynamics. It's these cumulative, kind of a cumulative Gaussian curve. This sort of curve with an inflection point, both an upward and a downward facing curve with an inflection point in the middle. Let's walk ourselves through this, shall we? And try to understand the basic saturation curve before I tell you how we're going to change this curve, how to affect this curve. So, along the bottom axis, we have partial pressure of oxygen, right? And along the y-axis, we have saturation of hemoglobin, right? Percent saturation of hemoglobin from 0 to 100%. And that is across the population of hemoglobin that you would find in an ensemble, okay? Because, of course, hemoglobin itself can only have one of 5 states, right? It can have fully deoxy, 1 out of 4, 2 out of 4, 3, or fully saturated, right? It only has 5 states. But these saturation curves are looking at ensembles of hemoglobin, all right? So right here along this axis, the partial pressure of oxygen, we have right about here, this is roughly, this 40 is referring to this line, this is roughly the concentration of the partial pressure of oxygen in your veins, okay? So this is the concentration of oxygen at the peripheral tissues, whatever tissues, this is venous blood. And at that partial pressure of oxygen, when you are at rest, of course this is at rest, then you're going to have roughly 75%, roughly 75% saturation. Yes, this does mean that there are going to be some hemoglobin at this partial pressure that are fully saturated. There are going to be some that are at 50% saturation. But on average, in the ensemble, it's going to be 75% saturation, okay? So for every 100, there's going to be a 50, etc. Or for every 100, there's going to be some balance of the things on the other end. But in average, the vast majority of the hemoglobin are going to be at about 75% saturation, okay? Now, this blood is taken over to the lungs, okay? And we can see that up at the top up there. At the lungs, the partial pressure with arterial blood is around 100 millimeters of mercury. I showed that on the first slide at the beginning of lecture today, right? So here, at this point, we have roughly full saturation. It shows it going to 100. It's a little bit, I have it saying greater than 100. Oh, I see. Yeah. I have it at 100%. It's not actually 100%. It's something like 98% saturation. Okay. There is going to be a small number that are at 75%, for example. But it's very close to 100% saturation when you get up here around 100 millimeters of mercury. So the hemoglobin picks up the oxygen, and then it goes through the circulatory system, gets brought back down to the tissues, and unloads that oxygen. You'll notice that this curve has an unusual shape. All right? This is due to the fact that the binding of oxygen facilitates the binding of more oxygen. The fact that it's not a straight line, for example, means that there is something that's changing the facility with which oxygen is binding to hemoglobin. All right, this is what we call allosteria. So when you are at rest, up to 3/4 of the dissolved oxygen in your blood can be reserved by the red blood cells. So you're at rest, and this is venous blood. 3/4 of the oxygen is not coming off of that hemoglobin. It's just staying there. It's just staying there. We're only ever trading one off of one of the four sites. Why? What's the advantage to this? Yeah.
It makes it easier to bind to more oxygen since they're keeping an oxygen. If they unloaded all of the oxygen, then it would be harder to.
Okay. But then why do we have this tetramer? Well, I'm asking, you're correct, but why do we have this reserve of oxygen? Couldn't we just have a single molecule, like myoglobin is a single molecule, that goes, picks up some oxygen, and then dumps it over here, and then comes back? Yeah.
Well, you need the multiple subunits to be able to have that allosteric effect.
Yeah, but how is the allosteric effect even helping us yet? I'm not quite sure how it's helping us yet.
Prepared for exercise?
It prepares us for increased oxygen demand, right? If we suddenly have a problem where we're down here, or we fall under the ice and we're down here, and we don't have access to oxygen, we go into a different part of the curve where the slope of that curve is totally different than the slope of this curve up here. Totally different than the slope of this curve. Okay? We suddenly go from the shallow part of the curve to, oh wow, we need oxygen. Steep part of the curve. We're going to unload it rapidly. We're going to unload it rapidly for a relatively small change in the partial pressure of oxygen. So for a big change in the partial pressure of oxygen, we don't unload that much gas. But so we're going to saturation. I'm sorry. We're going 20 millimeters of mercury change in the partial pressure of oxygen in this direction, and this is how much oxygen we unload. Compare that to 2 changes in partial pressure of oxygen over here, and we unload a significantly greater proportion of that oxygen. All right? So allosteria helps us when there is increased oxygen demand, when you're having a problem, whether you're exercising or have some sort of exertion, or whether for some other reason you have had oxygen depletion. Okay? So it's an emergency backup. Those 3 oxygens are reserved. And this is where allosteria really comes into effect. It's giving us different slopes of the curve for different regimes of partial pressure of oxygen. All right? Different slope of the curve in here versus down here, if we ever have to dip into this part of the curve. And we'll see how we do in a moment. Yeah, you're looking skeptical, Haley. No? You got it? All right. Here we go. So here we are at 40 millimeters of mercury. And here we are at the lungs. About 21% of that oxygen that's being carried by hemoglobin, 21% of the oxygen carrying capacity is being offloaded at any one given time. All right? However, let's exercise, let's put some demand on the system, and suddenly there's another 45% of that oxygen that we can begin to access because we are in a much steeper part of the curve here, for a change of only 20 millimeters of mercury. Whereas going from here to here, this is a change of 60 millimeters of mercury, and we're only getting 21% of the oxygen off? Because you're sitting there listening to me talk. You're not running away from werewolves. I have to always avoid political jokes. I just laugh at them in my head. So,
One can imagine.
yeah, 45. Yeah, insert your own joke. Are there questions on that? We're going to see how this plays out in other ways in a moment. All right. So it turns out, I talked about this conformational shift. We call these conformational shifts the T state and the R state. The tense state and the relaxed state. It turns out that hemoglobin can actually be in the T state or the R state at any point in saturation. When I started first showing you those GIFs of shifting between the T and the R state, right, I was associating that with oxy and deoxy, but it's a little bit more complicated than that. It can be in one or the other. These are equilibria between those two states. All right. It just so happens that when it's deoxy, the T state is strongly favored. It probably is in the T state when it is deoxy, but it can be in the R state. It is an equilibrium. It's just a strong equilibrium in the T state direction. On the other hand, the R state is strongly favored on when it's in oxyhemoglobin. All right. Did I say T and R stand for tight and relaxed? Did I say that already? Okay. We're going to see what this means in a little bit. And in between, you can see here at 50% saturation in a hemoglobin that it's a roughly balanced equilibria between the two states. Okay. It gets more sophisticated than this even. This is a bit of a simplification. Hemoglobin, there was, when I was in graduate school, the kineticist in the department at Notre Dame taught an entire class about kinetics, talking almost exclusively about hemoglobin. It's a very sophisticated molecule, actually. But we're just scratching the surface of it. So if you were to be frozen in either the T state or frozen in the R state and not able to interconvert between the two of them, you would have a T state would look like this, roughly linear. And the R state would have this binding curve that looks very similar to the binding curve of myoglobin. If I were to, so myoglobin is a, did I talk about myoglobin when we were talking about muscles? How myoglobin is the oxygen-carrying globin that's in the slow oxidative muscle fibers, and it's a monomer that is not allosteric. All right. So the observed hemoglobin binding curve is this one in the middle. It's sort of a combination of the two and reflects this spectrum of equilibria across the saturation profile of hemoglobin. All right. So now is the story, and I wish I had more time because this is like the coolest thing. I'm going to introduce 2,3-BPG, bisphosphoglycerate, and this is the introduction so I can begin to talk about fetal hemoglobin, which is the coolest story that I think I have to tell. So 2,3-bisphosphoglycerate. I guess the first story I will say is this. If I were to purify hemoglobin from the blood, and this was the original experiments made this observation. This is how they discovered the role of 2,3-BPG. If you were to purify hemoglobin from the blood and look at the oxygen saturation dynamics, so just expose it to an increasing level of oxygen and look at the percent saturation and plot this curve out, you would see this curve in black here. And if you were to compare the exercise concentration of oxygen, so that's 20 millimeters of mercury, right? So that's pretty much the maximal demand, barring an emergency, the maximal demand that your body's going to have. It'll, in the tissues would be maybe down to 20. Going all the way up to the 100 at the lungs, pure hemoglobin only offloads about 8% of its oxygen. So you're thinking, wow, you just lied to me. You told me it was 21%. Or no, it was 66%. It was the 21 plus the 45. Right? This slide back here. I just told you that it was 2/3 of the oxygen got unloaded here by hemoglobin when I'm exercising. What's going on? Is this the era of alternative facts in the classroom? No, it's not. We have hemoglobin in red blood cells does perform the way I showed you, where we have this 2/3 of the oxygen being transported during exercise from the 20 torr up to the 100 torr. Why? What's different? Well, in your cells, there's this intermediate in glycolysis 2,3-bisphosphoglycerate. It's basically a 3-carbon derivative of pyruvate that has 2 phosphate groups on it. The presence of 2,3-BPG gives us this curve that we see here. All right, so it's some kind of an effector. This is some kind of molecular effector. In fact, you will see that it helps to stabilize the T state and shifts us from that R state curve I showed you a little bit closer to the T state curve. All right, so without 2,3-BPG, we are pretty close to the R state curve. In the presence of 2,3-BPG, it shifts us down to this observed binding curve, which is a little bit closer to the T state. This is because 2,3-BPG stabilizes the T state. Well, Dr. K, how does this happen? All right. It happens because of the donut hole. 2,3-BPG carries 5 charges, 5 negative charges. 2 from each of the phosphates and 1 on the carboxylic acid, the carboxylate anion on the glycerate. All right? 5 negative charges. In this little donut hole here, we have on the beta-1 subunit, we have a histidine 143, lysine 82, and histidine 2. Those are all positive, those are cations, as well as on the beta-2 subunit. There are 3 cations. So this is a +6 charge pocket inside that donut. And 2,3-BPG sticks itself in there and kind of locks it in. It kind of locks it in to that orientation a little bit because of charge-charge interaction. You guys follow this? This has implications for fetal hemoglobin, and then I'm gonna let you go after this point. So in fetal hemoglobin, histidine 143 is mutated and is serine. That plus charge on the nitrogen here in histidine is now replaced by a hydroxyl group in the serine. likewise in the beta 2 subunit. All right? So we have reduced the positive charge from 6 to 4 in this pocket in fetal hemoglobin. Not embryos, but unborn babies in the mother's womb. Okay? So 2,3-BPG has a lower affinity for sticking into that binding pocket. Okay.